Good evening, ladies and gentlemen. If I can have your attention, welcome to the lecture given by the winner of the Peter Wildy Prize for Microbiology Education. This is awarded annually for an outstanding contribution to microbiology education. And this year's prize winner is David Bhella from the MRC Centre for Virology Research in Glasgow. He's been recognised for his excellent outreach programme, particularly with enthusing school children, about careers in science and especially microbiology, and also for his other public engagement activities. Now, David works in Glasgow. He works quite closely with the Glasgow Science Centre. And for those of you who know anything about the popular politics of the central belt of Scotland will know that for somebody who comes from Edinburgh, as I do, it's actually very difficult to say anything positive about Glasgow. But I will say very positive things about David and about the Glasgow Science Centre. If you're ever north of the border, you really must visit it. Now, this talk has been live streamed, so I'd like to welcome you who are in the room. I'd like to welcome those of you who are in the rest of the UK, the rest of Europe, in the Americas, and for the insomniacs in the rest of the world. With that, I want to do no more interference with what we're here to do, which is to listen to David give his talk on beautiful and a little bit scary viruses and science communication. David Bhella.
Thank you very much. I should like to start my talk today by thanking the SGM for this wonderful honour and this wonderful opportunity to talk about the outreach programme that I've developed in Glasgow over the last decade or so. So there are two really strong influences in my life that have directed my career towards being engaged with science communication and outreach. And I'll leave it to you to decide which of these two key influences in my life are best described by this title that I've applied for my talk, beautiful and a little bit scary. So I'm going to start my talk today with a little bit of biography information about myself and how I became involved in public engagement and outreach. I shall then go on to talk a little bit about a schools workshop that we developed in partnership with Glasgow Science Centre to teach school children about molecular biology. I'll then say a little bit about Meet the Expert events, which are an ideal opportunity for us to become engaged in science communication without perhaps quite so much investment of energy than is required for such activities as the DNA Technology Workshop. I'll then move on to talk about another schools event that we developed as part of the Glasgow Science Festival, and then say something about art science projects with specific reference to an exhibition that we created called Molecular Machines. I'll then sum up my talk with my thoughts on working with science centres, festivals and museums, and a little question at the end here about what we can do with outreach. What are the benefits of outreach for ourselves, and can outreach activities change the lives of our audience? So I'm going to start with a little bit of biographical information about myself. My first degree was in biology, and shortly after graduating, I was looking for a job that was involving electron microscopy, which is something I did in the course of my degree and I really enjoyed. And this opportunity arose to become a trainee medical laboratory scientist working in the Royal London Hospital in the East End of London, where principal amongst my responsibilities was to perform diagnostic electron microscopy for viruses. And it was at this time that I was really struck by the beauty of viruses and how amazing they are to look at in the electron microscope. And every time I got a new virus into the electron microscope, I'd get really excited and spend the next hour or so printing my micrographs in the darkroom, and then before rushing home to show them to all my friends, much to their amusement. So this is sort of my first exposure to virus structure, and it inspired me then to move on to pursue an academic career in virus structure research. So I entered for a Ph.D. in Birkbeck College, working on cryo-electron microscopy and structure analysis of retrotransposon capsids. This was working with Professor Helen Sable in the crystallography department. So cryo-electron microscopy is a really nice technique for looking at the structure of large macromolecular assemblies. They can be imaged in the electron microscope in a frozen hydrated state, and then we can use computational image reconstruction methods to extract structured data from these images. When I started working in cryo-electron microscopy, the resolution was quite low, but in the intervening years, electron microscopy has evolved into a structural biology technique that can now challenge crystallography in terms of the resolution that can be achieved. We can achieve close to atomic resolution structure data on subjects such as viruses. So after completing my PhD, I then moved to Glasgow to set up our cryo-EM research group, and I worked on many virus systems since then, and using cryo-EM to look not only at the structure of viruses, but also to look at interactions between viruses and ligands that can bind to the capsids. So these could be virus-host interactions, such as the two structures I'm showing here, which are viruses decorated with soluble fragments of their cellular receptor. We can then dock crystallographic data or high-resolution data into these maps to build what's called a quasi-atomic resolution model of the virus receptor complex. I hope you can see from these animations that something I take a great deal of pleasure in is in developing really eye-catching imagery from these already very beautiful structures. So I made these animations using a macromolecular software package called Chimera, which is has very powerful tools for animation and visualization of structure. And this is something I really enjoyed. And in showing images like this to my friends, a lot of my friends would then comment that this is really eye-catching and this could be a really engaging thing that could be turned into an art exhibition. And that's actually what happened and led to the development of our Molecular Machines exhibition. But I also became interested in going beyond the sort of capacity of macromolecular structure visualization software to using commercial 3D animation software to try and tell the story underlying the biology of the research projects that we were engaged with. So I'm just going to show a short movie now that I developed using an animation package called Maya, which shows the structure of adenovirus in the bloodstream as it becomes decorated with blood coagulation factor 10. This is some work I did in collaboration with Andy Baker in the University of Glasgow. An adenovirus, when it's delivered intravenously, becomes quickly coated in factor 10, and this leads to the virus being sequestered in the liver, which of course is a problem if you're interested in using this virus for gene therapy. So I've started developing my skills as an animator and using these techniques to try and tell the story of the biology of our research. But I also then started using this software to develop teaching resources for my practice as a scientist. So when I give a research seminar, I have animations now for teaching people about cryo-electron microscopy, and also in our public engagement work. So this is just a quick show of an animation, a short snip of an animation that I developed on cryo-electron microscopy. And this particular clip shows how we use electron tomography to analyze pleomorphic viruses in the electron microscope by tilting the specimen in the electron microscope and then rotating it in the electron beam and recording images every 1 or 2 degrees. So these images on the right here are filamentous influenza virus. So I've become very interested in digital media and using digital media as a teaching tool. Another key influence in my life is in my becoming involved in public engagement and outreach activity. Another key influence in my life generally is my wife, who in 2003 became a part of the science learning team in Glasgow Science Centre. So Glasgow Science Centre is a Millennium Project museum. It opened in June 2001, and it occupies this iconic building at Pacific Quay on the banks of the River Clyde in Glasgow in the Govan district. And here we see it on a typical Glasgow summer's day. Glorious. So Glasgow Science Centre has about 250,000 visitors a year. It has 3 floors of interactive exhibits, but it also has a very large programme of workshops and activities for both public and schools audiences. And these workshops, developing these workshops, is actually what my wife does in Glasgow Science Centre. So shortly after she joined GSC, she started to ask me whether we could work together to develop a workshop to teach molecular biology, in particular the polymerase chain reaction for students at the high school level in our area. So this is something that schools really struggle to teach. They have a lack of equipment and expertise to effectively teach molecular biology. So by creating a workshop, we hope to attract students to Glasgow Science Centre and actually provide something that would be genuinely valued by the education community in Scotland. So we developed this activity, and we first ran it in 2005, and we've run it every year since then. And every year, until last year, we ran it for 4 days a year, a full one-day workshop for school pupils. Last year, we actually expanded it for 6 days a year, and I hope that we'll continue to be able to deliver this at that level. And every day, we have 24 students and their teachers come to Glasgow Science Centre to take part in this workshop. So over the years, we've seen over 700 students and their teachers. So the workshop is staffed by quite a large team. We have one member of the Glasgow Science Centre Science Learning team, as well as myself, and also my technician, Colin Loney. And we also take a Ph.D. student and a postdoc from the CVR every day. And I think this is really important, that the students are exposed to people at different career stages and playing different roles in the academic research lab. There may become a time in the course of a workshop when a student may want to ask a question, but may be a little bit intimidated about asking the program leader that day. So a PhD student's a little bit closer to them in age and maybe a little bit more approachable. So I'm just going to work my way through the course of the workshop now and what we do in that activity. So in the morning, we start our workshop with some introductory talks. And I think this is very important to actually make sure that everyone's on the same page. So we start out with a little bit of revision about DNA and the structure of DNA, and then a little bit of teaching about the polymerase chain reaction and how it works. So when developing these talks, I was really, in the first instance, I just set out to try and find some animations on the internet. There are some really beautiful animations of PCR and DNA structure on the internet, and I looked into whether we could exploit these in our introductory talks, but it quickly became apparent that these sort of movies that were available weren't flexible enough to use as teaching tools. You couldn't stop and start them, you couldn't focus on specific areas of interest. So this was actually one of the first animations I developed in Maya. I developed a series of animations, first of all looking at the structure of DNA. So I always think it's really important, something I've learned from structural biology is if you're trying to look at a complex macromolecular structure, it's much easier to understand the structure if it's actually moving in a slide presentation such as this. So straight away we get some understanding of the nature of the DNA structure. But also we can modify our animations to teach specific aspects, so we can color the structure to specifically focus on the nucleotides and teach about base pairing in DNA structure. And we can also dissect the molecule to focus on specific structural features that we're interested in. Here we're just looking at the purines and pyrimidines and the hydrogen bonding that occurs between these two classes of nucleotide. So once we've gone over DNA structure, we then begin with some teaching on PCR and how PCR works. And again, using some very simple animations. I have in the top left corner here a thermometer which tells you the relative temperature. And we're interested in our PCR experiment in amplifying this specific sequence of DNA. So I'm just going to go through one cycle of the PCR using this animation. So we heat the DNA up, the DNA strands melt, separate. We then have primers that anneal. And finally, the Taq polymerase comes along and elongates the strands. And I'm sure I don't need to teach all of you about PCR, but I'm just illustrating the animations that we've developed to help us explain to students how PCR works. So in the actual presentation, we work through this by another couple of cycles of PCR showing how we get an exponential increase in the quantities of DNA present. After we've gone through this initial teaching, we then go on to introduce the experimental protocol. So one thing that I think is really important when you're trying to teach students something that's quite complicated is to put that experimental protocol into some sort of context. And I think it makes it really much easier for the student to learn what they're doing if they understand why they're doing it. So for this activity, we have a very, very simple context for our experiment, and that is that we're doing a diagnostic PCR to diagnose the cause of facial vesicular rash in 2 patients who are immune compromised. And the potential cause of this infections are either the herpes simplex virus or varicella zoster virus. So the experiment that we actually do is a diagnostic PCR to detect whether herpes simplex virus or VZV in patient samples from these 2 patients. So, of course, we tell the students that it's extracted DNA from swabs taken from these patient samples, but I think GSC may have issues with us bringing real virus into the laboratory. So actually what we do is just a PCR using plasmid DNA. So once we've introduced the patients and told the students about the experimental protocol, we then go through some pipette practice using the micropipette, so the students are able to work with the tools that we've given them, and then they set up their PCR reaction that morning. So this brings us up to the late morning. The PCR reaction has been set up and put in the thermocycler, and while that PCR reaction is going on, we then have a little think about careers in biomedical research. So again, this is just a short talk on the different career options for people in academic research. And I think what I really try to put across in these talks is not, it's really important that everyone becomes a scientist, but is to try and put across the excitement of being a scientist. And actually in all the work we do is to try, the key message is how exciting it is to be a scientist, and the excitement that we get from the process of discovery, from being the first person in the world to understand something that we've been working on, which I find particularly gratifying, and I expect most of you do as well. So we have a talk about careers in biomedical research, looking at life in an academic research lab, the various roles and responsibilities that there are. And then we have a talk from our Ph.D. student who's along with us that day, and they give a talk about their career and their project, a little perspective on their career development and some of their ambitions for their career. We also then broaden the conversation to include a talk about alternative science careers and also jobs that are available with transferable skills if you were to undertake a science degree. So these are non-science jobs that would still benefit from having exposure to the transferable skills that you get from a science degree. So the students then stop for a lunch break, and after lunch they return to the lab, and we get on with the experimental protocol for the day. So the PCR will have run, so the students get their PCR reactions back, and these are then loaded onto agarose gels for electrophoresis to detect the DNA products of their PCR experiments. And while those DNA gels are running, we then have another taught activity looking at DNA sequencing, specifically Sanger sequencing. So when I first developed this activity with colleagues in the CVR and with the Glasgow Science Centre, it was my intention to develop another series of animations to teach DNA sequencing. But as the workshop came closer and closer, I realised I didn't actually have the time to develop those animations. And at the very last minute, a member of the science learning team came up with this activity, where instead of having a high-tech digital animation, we have laminated cards with nucleotide letters on that just have a magnet on the back that stick onto the whiteboard. And actually it works so well, I've never seen any reason to change it. So, as I say, we have the sequence that we're interested in trying to analyze. We have a pool of nucleotides and dideoxynucleotides that are fluorescently labeled. And this is Susan Meekleham from Glasgow Science Centre. She's one of the science learning team, and she's going to just explain a little bit about how DNA sequencing works for just 2 seconds in a movie.
So, DNA polymerase adds nucleotides on to this growing strand here. So the first nucleotide would be a T, because it's complementary to this A here. And then DNA polymerase may pick up a dideoxy nucleotide this time. So let's say it picks up a dideoxy T, and we know that that's the end of that growing strand. So this strand of DNA cannot elongate any further.
So what's really nice about this activity is that it's very simple, it's very low tech, but when Susan delivers it, she asks the students to shout out, what's the next nucleotide, what's the next nucleotide, and it gets the whole group engaged in it. It's not anything challenging for them, and everyone's quite happy to shout out what the next nucleotide is going to be. So Susan then goes on to explain how this is cycled to produce differently sized fragments that are fluorescently tagged, and how by putting these through a gel or a column we can then produce a chromatogram which can be read to generate the DNA sequence that we're interested in. So to consolidate that learning, we then give the students a real sequencing chromatogram that was actually generated in my lab a few years ago. And what we've done is deleted 21 nucleotides from the sequence. And we asked the students to just fill in that missing sequence. And this sort of consolidates their learning, helps them understand really how DNA sequencing chromatograms are and how they work. But this sequence is then taken, they write down the sequence that's filled in there, and they then enter that into the BLAST database. And this reveals that this sequence comes from the measles virus. We then go on to discuss a little bit about the chromatogram and the origin of that data. And that's very important, I think, because it exposes them to real research data, and we then provide the context by explaining to them the experiment that we were trying to do when we did this DNA sequencing experiment. Okay, So the last stage, the last sort of talk component, or the last discussion component of the workshop is a debate. And this is sort of quite an interesting, I think this is the most fun bit of the workshop for the facilitators, for the people who are organising the workshop. And it addresses a new part of the Scottish curriculum called Topical Science, which is all about dealing with the ethics of science. And it requires a different approach to teaching. It requires us to sort of facilitate discussion and allow students to express their own views rather than simply imparting knowledge to them. And it's very important when teaching ethics that the students should understand that there's not necessarily a right answer. So when we do this workshop, the students are divided up into small groups. So we have 6 pupils with 1 facilitator at each table in the lab in GSC. And we ask them to consider the question, if the UK government were to sequence the genomes of the whole population, who should have access to your data? And what we do is we give the students a number of cards with different possible candidates for people or groups of people who might wish to have access to those data. And we ask the students then to consider who of these should have access to that data. So we deal those cards out, so every student at the table will have 1 or 2 of these cards, and we ask them to think for themselves, just to take a little bit of time to themselves, to have a little think, and come up with their own ideas about whether or not these groups of people should have access to genetic sequence data on us, on the population. We'll then ask them to pair up with their neighbour, someone sitting close to them, and talk about their ideas a little bit further and maybe moderate their thinking, maybe come up with some new ideas, some new arguments. And then finally we ask them to share their thoughts on this question with the whole group. And we do this by placing the cards on a continuum between strongly disagree and strongly agree that a specific group or person or group of people should have access to this genetic information. So this think, pair, share approach is a really powerful way of getting people, everyone in the group, taking part in the activity, talking and contributing to the discussion. And it works very well in small groups. But think, pair, share can also work really well in quite large groups. And so to try and as an experiment to sort of test this hypothesis, I thought it would be kind of fun if we did a little bit of think, pair, share here today. So what I'd like you to do is just take a few seconds to have a little think about the question that if the UK government were to sequence the genomes of the whole population of the UK, who should have access to your data? And I'd like you to just think about pharmaceutical companies. Do you think pharmaceutical companies should have access to that information? So if you just take a little bit of time to have a little think to yourself, and then once you've made up your mind, talk to the person sitting next to you and see if you can come up with a view whether you agree or disagree that pharmaceutical companies should have access to this information. And we'll just think about that for 30 seconds and then we'll come back. Okay. Wow, okay. That certainly elicited some very animated conversation in the room, didn't it? Okay, so it would be great actually to be able to canvass everyone's view on this and have a proper discussion, but unfortunately I have to move on. So maybe what we could do is just have a show of hands for how many people agree that pharmaceutical companies should have access to this genetic sequence information. We have a show of hands. Okay, good. All right, so that was maybe about 15 or 20% of the audience. Does that reflect the 15 or 20% of the people that work in the pharmaceutical industry, perhaps? Or maybe those that don't. Yeah, I don't know. Okay, so what we do with the students is we actually have a very animated conversation. And the students are encouraged to take one of their cards and place it on this continuum. So, for example, they might strongly disagree with the idea that the government should have access to our genetic sequence information because they would probably use it for some hideous, nefarious process of genetic cleansing or something like that. But then what we do is then we open that conversation up to the rest of the students and ask them to sort of try and pose a counter-argument. And sometimes it may happen that all the students have a very similar opinion. And so then the facilitator might then pose an alternative argument that actually, well, the government has to do a lot of budgetary planning for healthcare. Maybe it would be really beneficial if the government had perhaps anonymized data so that they knew how many people were liable to develop a specific genetic disorder. And after this discussion, we may then moderate our view and move it along on the continuum. And we would do this for all of these different groups, and we end up with sometimes some really surprising answers and contradictions in the viewpoints at the end of the day. So this debate is really good fun. Everyone who takes part in it really enjoys it. The facilitators, the students from the CVR who come along and participate really enjoy this aspect of the day. And as the debate wears on, and maybe we feel we've exhausted all the sort of questions, there's room for that conversation to then move into other areas. Perhaps the students can ask the facilitator about their career, about careers in molecular biology research or something like that. And so this sort of naturally winds up the day with a nice discussion of what we've done that day. And the last thing we want to do then is then actually have a look at the results of our experiments. So we do a real PCR experiment. There's nothing fixed here. So the students have to actually be quite diligent in their pipetting, and they have to make sure they haven't cross-contaminated their experiments. And I think this is a really important part of the workshop, to make sure that we're doing a real scientific experiment and providing a real scientific experience. And one of the outcomes of that is that quite often a proportion of the reactions don't work. And this is really important. This is important teaching and learning because it's very important that students should understand that real science is like this. Sometimes your experiments don't work. And actually allows us to then go back through the course of the whole day, talk about the experiment, talk about the controls, talk about everything they've done, and say, well, where do you think in the course of the day maybe we might have made a mistake? Perhaps there was a pipetting error. Perhaps one of the controls cross-contaminated our reaction. So in going over the day's experiments and looking for these points where the mistakes can take place, this elicits a deeper understanding for the students. And it also allows the student to build a personal narrative of their experience in doing this workshop. So they might go away from the workshop saying, great, I got a really beautiful gel, all the bands were fantastic, but my pal, he really screwed it up. Yeah. So, but it allows them to learn from their experience, and it becomes a very much more personal thing for them if they discuss this aspect of the research, of the work that they've done. Okay, so whenever you do an activity such as this, it's really important to evaluate your outcomes, to evaluate the workshop and make sure that it's as good as it can be. So we always go through a process of evaluation by asking the students to complete a form saying how the workshop has been for them. And generally they're very, very positive about the activity. I'd just like to draw your attention to this bottom line here. It made me consider biological science as a career. 57% of them agree, 43% neutral or didn't answer. Now that might seem like quite a low percentage actually, but I'd like to point out these are advanced higher biology students. They're 16 to 17 years old. Many of them have already got quite a strong idea of what they want to do at university. So a lot of the sort of feedback that we get in the written comments is that, well, I really enjoyed the workshop, but I've already decided that I'm going to go and do pharmacy or dentistry or medicine or law. So actually in that context of students already having a strong idea of what they want to do, I actually think 57% is not bad. It's a pretty high percentage of students who maybe hadn't considered before now saying, well, actually, yeah, that was really good, and I am going to consider science as a career, biological sciences as a career. So looking at other outcomes from this workshop, the workshop was cited as an exemplar by Her Majesty's Inspector of Education. So HMIE is the Scottish equivalent of Ofsted, the Office for Standards in Education. In 2011, it merged with Learning Teaching Scotland to become Education Scotland, which is the main quality assurance agency in Scotland for its education. And Education Scotland also more recently adopted our teaching resources for teaching PCR and DNA structure for school students. So these resources were made available on the internet for teachers to download to allow them to teach their school pupils about PCR and DNA technology. Finally, a sort of a more general statement is that schools really value the activity very highly. And just to give you some evidence to support that suggestion, I'm just going to show you how far schools are prepared to travel with their pupils to participate in the PCR workshop at Glasgow Science Centre. So this is Scotland. This is what it looks like right now, covered in snow. And this is Glasgow, which is also covered in snow, actually. And most of our students actually do come from the greater Glasgow area to participate in the PCR workshop, but we do have groups come from further afield. We have groups come from Ayrshire, from the Firth of Clyde. We have groups coming from the Clyde Valley in Edinburgh, and from further afield still up in Fife, down into the borders. We have groups come from down in Dumfries and Galloway and Kirkcudbright, and we also have groups coming from the Highlands. So we have classes regularly attending from Culloden, which is near Inverness. So that's groups of teachers who are prepared to travel 170 miles with their student to come to Glasgow to participate in the PCR workshop. So I think that speaks volumes, actually, that it really is highly valued by teachers in Scotland. So the PCR workshop I've told you about is something that was a really big investment in time for us. We put a lot of effort into developing the activity, and every year we spend a lot of time making the reagents and delivering the event. So Meet the Expert activities are something that I would encourage all you to consider becoming involved in. Meet the Expert events will take place in most science centres, science festivals and museums, and it really provides a great opportunity to become involved in outreach with all the support of those organisations without necessarily having to make all the investment yourself. And I'd just like to say, when you do something like a Meet the Expert activity, it's very tempting to focus on what you do and trying to teach something and your message being something very rooted in what you do. But that's not necessarily essential. I think the main thing is to engage young people in these activities that relate to biology. So we can do activities that last maybe 10 to 15 minutes, and they don't always have to be about virology or microbiology. We can do something very simple that you probably all will have done at one stage, something like extracting DNA from fruit. So this is an activity we actually did as a Meet the Expert activity to support an exhibition that opened at Glasgow Science Centre, which was funded by the Wellcome Trust, called Inside DNA. So we just came along and did this it's DNA extraction from strawberries with some very, very young children. And the main thing is to provide exposure to a real scientific experiment, to use some real scientific equipment, and use this as a hook to engage the young audience and talk to them about anything they want to talk about. So they can have that opportunity to speak to an expert about scientific research, about anything at all they're interested in. And this also provides the opportunity to speak to these children's parents, who are the main key influencer in those child career choices. So it's a great opportunity to really have a great depth of engagement, and it doesn't have to always be about that narrative that you're interested in putting across. We do have activities that are related to virology, Meet the Expert activities. We have a microscopy of viral cultures. So we prepared some cell cultures, infected them with herpes simplex virus and measles virus, and then we formaldehyde fixed them and sterilized them so that we could take them out and show them to a public audience under an inverted microscope. And again, this is really nice for the audience to really start using real scientific equipment. It's very, very engaging for them to look down a real microscope at something and have you explain to them what they're looking at. We do have another activity called Build a Virus, which was actually developed not by myself, it was developed by the MRC Corporate Communications team, originally for the Edinburgh Science Festival. I have to say, initially when I heard about this activity, I wasn't that keen, because I fairly strongly believe that we should be using science to engage children about science. But actually, having delivered this activity, I have to say it's really good. It has a great deal to offer. So what we do is we start with shredded bits of paper to represent a viral genome, and then we use air dough, which is like a kind of Play-Doh that hardens, to make a capsid around that genome. And then we use gaming pegs to represent the surface glycoproteins of a virus, and we push these gaming pegs into this model of the virus. And this allows us to talk to some very, very young children about some surprisingly complex concepts. Specifically, we can talk about the concept of a genome as a set of instructions for making more viruses. Having said that, we can then say, well, the genome is very, very important, it's very precious, the virus has to protect it by packaging it up in this capsid. And we can then start to talk about the fact that viruses are intracellular parasites by saying that we have these spikes on the surface of the virus, which are important for attaching to the surface of the cell that the virus is infecting. So from very, very young children, we can talk to them about some quite challenging ideas. So the last Meet the Expert activity that I'm going to mention now is Diagnosing Viral Diarrhea. Again, hugely engaging. So this is actually a mock latex agglutination test. It was developed by Christine Peters, who's a clinical microbiologist in Ayrshire, and she shared it with us actually for the Glasgow Science Festival, but we've used it as a Meet the Expert activity on several occasions, and I've just supported it with a little animation that I made that I show on an iPad to explain how latex agglutination works. So I'm going to say a little bit more about this activity in just a moment, so I won't expand on it just now. So we have all these Meet the Expert activities, and these all came together actually when we were asked to develop an activity for the Glasgow Science Festival. So Glasgow Science Festival happens in Glasgow every summer. It's organised by Glasgow University, and it has a whole load of activities that are oriented towards public and schools audiences. So we were asked to participate in a microbiology-based activity for school students in this age group, 16 to 17 years, that would last about 2 hours. And we did this in partnership with scientists from the Wellcome Trust Centre for Molecular Parasitology, which is also in the University of Glasgow. So we were thinking about what we could do, and we had all these activities that we'd already used or in past science festivals or in Meet the Expert activities. We had this mock latex agglutination test. We have fixed viral cultures that we can show the students. And what we wanted to do was come up with something that would expose the students to a little bit of molecular biology as well. But of course we couldn't do PCR because PCR would just take too long. So what we decided to do was just do a restriction enzyme digest of some DNA, and then run that digested DNA on an agarose gel to separate it according to size. So this is obviously a very nice thing to do, but we had to provide a context for it. As I said earlier, I think it's really important when you're trying to teach students something to actually put it in a context so that they can understand why they're doing it. So how do we tie these disparate group of experiments together, and how do we make them exciting, and how do we add educational value? And the way I did this, the way I attempted to do this, was by providing a very strong backstory that would hopefully be very engaging. And the backstory was one of an outbreak of a new and very frightening hemorrhagic disease virus called Jugu virus. So this fictional virus was discovered in the Democratic Republic of Congo. And in 90% of people who are afflicted with this virus, we have a self-limiting gastroenteritis that lasts about 48 hours. However, 10% of the people who are infected with this virus go on to develop a hemorrhagic disease, which is fatal in 90 to 95% of the cases. So this virus is very, very infectious. It's actually related to norovirus. And the patients, the other key aspect of this backstory was that the patients who developed hemorrhagic fever had been discovered to have a mutation in their interferon gamma gene. So this story was underpinning these very different experiments that we wanted the students to do. So we were going to use the latex agglutination test to look at a number of patients and determine whether they had the Jugu virus. We were then going to use restriction fragment length polymorphism analysis of the interferon genes from these patients to determine whether they were susceptible to the hemorrhagic disease by having this mutant interferon gamma gene. So we had 6 patients. These are the stool samples from those 6 patients, and each of these 6 patients also had a very strong backstory. For example, patient 6 was a member of Scottish Parliament who just returned from the Democratic Republic of Congo after meeting with the Kabila government on a fact-finding trip. I thought it was very important that one of the potential victims of this frightening, horrible virus should be a politician. So this achieved a high level of engagement with a teenage audience. So we have a strong story, a strong narrative of this very, very frightening virus outbreak, and we're trying to do some research and to try and find out whether some patients who have just returned from the Democratic Republic of Congo to Glasgow may be infected with this virus and whether they're likely to develop the hemorrhagic disease. So we have this strong story, and we also have these very engaging and very realistic looking fake poo samples, which are also, if there's one take-home message from this lecture, it's fake poo, very engaging. So actually these stool samples are made of Weetabix, food colour, water, and in order to make a positive sample we'd make the pH about 2, and the latex for this test is actually milk. So when you mix milk with a liquid at pH 2, it curdles and it looks exactly like agglutinated latex in a latex agglutination test. So the students would take these stool samples, they would take a little bit, mix it in a buffer, vortex it, centrifuge it, and then mix the supernatant with the latex to get this agglutination result. So the milk curdles at low pH, so that's what gives us this agglutination. They then did a restriction digest on some DNA, which we said was the PCR products for the interferon genes for each of these patients. And they digested that with the restriction enzyme and ran it on a gel. Because we were quite short for time, we use this flash gel system from Lonza, which is very, very fast. Because it has a transilluminator built in, it actually allows the students to watch the DNA bands as they resolve. So that's very interesting as well. So on the left we have the variant interferon gene and the normal interferon gene, and we can see that patients 2 and 4 also have this variant interferon gene and therefore susceptible to succumbing to this hemorrhagic disease. So there are a number of learning outcomes from this activity. First of all, the students have been exposed to molecular biology. We've taught them about PCR. They've done an experiment with restriction enzymes and gel electrophoresis. We've then talked about diagnostic methods, direct antigen detection. And finally, we've added in some current topics in virology. So we've exposed the students to some rather interesting hot topics in virology, particularly concepts of innate immunity, the importance of host factors in disease severity, and also concepts of personalized medicine based on genome analysis. So in our feedback on this workshop, we got very, very positive reactions from the students. In particular, they rated the learning about viruses and diseases very highly, and learning about diagnostics. And particularly, many of the students say they really enjoy doing the mock latex agglutination with the stool samples, having a scenario being something else that the students really enjoy. Okay, so I've talked a lot about schools, workshops, meet the expert activities, things that really require quite a large investment of time, quite a large investment of energy to achieve a level of engagement with a small audience that's very, very deep. So these activities allow us to have really good, long conversations with individual students, children, their parents, in a science centre environment. But of course, outreach projects can also be devised that reach much larger audiences with the potential to raise public awareness of microbiology research, and also to raise the profile of your own work and your own institution. So we saw this last year from Vincent Racaniello's talk, how public engagement activities can engage huge audiences using new media. So one activity that we developed to engage a larger audience was the Molecular Machines Images from Virus Research art exhibition. So this was also a close collaboration with Glasgow Science Centre, who, as well as supporting us by seeking funding from the Scottish Government, also provided the venue and arranged for that exhibition to tour other science centres in Scotland. And the last thing that the Glasgow Science Centre provided was a link with a very, very good artist who had lots of experience of working with science, and that artist was Murray Robertson, who's a very well-established artist in Glasgow. He's worked with the Royal Society of Chemists to develop a visual periodic table, and he's also worked with the Clinical Virology Network, some of whom may be here today, so they may know Murray. So Murray has a lot of really good ideas about ways of showing virus research data, and that was an important aspect of the Molecular Machines exhibition project, was that we wanted to make an exhibition from real data, and we wanted to engage the audience with real research data. So we generated a series of images based on research data, as well as a movie. And what we wanted this exhibition to deal with was a few key concepts in virology. Firstly, the concept of a virus as an intracellular parasite, the question of whether viruses are alive or not, and also to deal with questions of size and scale and structure when thinking about viruses. But the overarching thing that we really wanted to put across in this exhibition, again, was to try and convey that sense of wonder that's felt by research scientists during the process of discovery, and also to engender a sense of hope about the future for treatment of viruses through showing virus research in a very positive light, and showing the sort of techniques that can be brought to bear to address questions of virus disease. So Murray created these images by layering lots of different types of image data produced from virus research, including we have microarray data here. This is a modelled protein structure. We have a cryo-EM structure of cytomegalovirus. We have phylogenetic data, and in the very background there we also have a cell culture image. So we produced 14 of these images from research data, and we also produced an animation. So the animation was kind of an afterthought, but actually turned out to be one of the most successful aspects of the exhibition. And it had two components. The first component of the animation was to look at this replication cycle of a virus, focusing on herpes simplex virus. And the second series of animations was really just animations made from real research data, using confocal data, structural biology data, to make sort of more abstract animations that would be eye-catching and engaging for the audience. So I can't show you the whole animation because it's 8 minutes long, but I just wanted to show you just about a minute and a half of this animation from Molecular Machines. Okay, so the full animation can be seen on my YouTube channel and also on the Molecular Machines website. I'll give you the address for that at the end of my talk. So outcomes. So this is what the exhibition looked like. The exhibition launched in 2007 and was displayed in Glasgow Science Centre on a semi-permanent basis for quite a long period of time and in several different locations within the Science Mall. Actually, I thought the most successful location for the exhibition was actually as part of the queuing system for people wanting to buy tickets to access the Science Mall. And that's actually what we're showing here. So you can see these tensor barriers, which are the queuing system, but the exhibition was integrated into that, creating a captive audience where people were compelled to read it. I thought that was very successful and quite innovative of the Science Centre to come up with that. So this is a close-up image of the exhibition. I'd just like to sort of show you, we've got the images here and quite a bit of supporting text. And I think if I was going to do the activity again, I probably would reduce the amount of text that was on these exhibits, because having watched people interact with the exhibition, very, very, very few people would take the time to read all of this text. And sometimes I think it can be a little bit sort of overwhelming to see an exhibition with so much text on it. So now we have the opportunity to use QR codes to link to web pages so people can read this information using their smartphone if they're interested. So I probably, if I was doing this exhibition again, I'd reduce that. So the exhibition toured. It was reproduced in this large format for use at the Edinburgh Science Festival, and it also toured to Our Dynamic Earth in Edinburgh, Satosphere in Aberdeen, and Sensation in Dundee. These are the other members of the Scottish Science Centre network. And this proved to be a very successful event, and it was estimated that the audience was about 350,000, and it achieved considerable media coverage. First of all, it was reported on Scotland Today, which is our regional news bulletin, featuring an interview with both myself and Murray Robertson, the artist. This exhibition then led to the production of a 4-page article on the work of the MRC Virology unit, which was published in the Herald on Sunday, which is a broadsheet newspaper in Scotland with this lovely full-spread picture of myself in it as well, which was quite embarrassing and I didn't expect. The exhibition was also reported in the Evening Times, the Scotsman, and the Times Educational Supplement. And finally, it also led to a request for me to write an opinion piece on viruses for the Scotsman Science and the Environment section. So it achieved quite a lot of media coverage, and I think a lot of that was related to the fact that it was real science data, and it was showing viruses in this very sort of, as very beautiful, very structurally abstract sort of objects, and this was quite contradictory to what people might expect, people think of as what most people understand about what microbes look like comes from bleach adverts, and it's things with eyes and fangs and so showing viruses as really beautiful structures, I think, was quite surprising, and that led to this sort of strong media response. So this activity, and most of the activities I've talked about today, were done in partnership with Glasgow Science Centre or Glasgow Science Festival. So I'm just going to finish up now looking at what you can get from working with organisations like science centres. So science centres and festivals provide this structured route to outreach, and they provide a great support network of highly skilled staff who can provide you with, you and your team with extra training in workshop presentation skills. They have detailed knowledge of the curriculum and what the audience requirements are, and they can also help you with evaluation and marketing of your projects. But of course the main thing we get from museums and science centres is actually an audience, a ready-made audience who are there. So we have an audience of schools, families, adult learners, and we also have the science centres, for us, have also taken care of booking and publicity for all these activities. So that makes it a lot easier to do an engagement activity with a large audience and a great depth of engagement. So when working with science centres, we also have to consider what they want. So the Science Centre mission is to create a more scientifically literate society by acting as a conduit for these different agencies to come together and present their work and present science in a positive light to a public and schools audiences. So what they want from us is they want experts, they want real-life scientists who can come along and stimulate audience interest in science and impart their sense of wonder and their enthusiasm for scientific research. So you can do this through developing Meet the Expert activities. You can do this by developing more detailed workshops, art science exhibitions. But perhaps that's not for everyone. So you don't always have to actually be at the front line delivering data, delivering these activities. You can also work with science centres in an advisory capacity to help them deliver their mission, to help them develop activities. But if you can deliver exciting events and activities, so much the better. So I'm going to say a little bit now about the benefits of outreach activities. Quite recently, Research Councils UK published a pamphlet on the benefits of public engagement that had a number of case studies and came up with, at the end of this document, a long list of all the potential benefits of being involved in public engagement activities. And I really sort of wholeheartedly agree with all of these assertions, in particular skills development. So in all of the activities we do, the students who come along and participate, the postdocs who participate, get exposure to public engagement, and that becomes something they really enjoy. It becomes part of their own career. And it's good for their CV. It's also good for funding, if you're writing a grant application, to be able to say that you're doing these sorts of activities. But what's really good for is enhancing your profile and the profile of your institute, both within the scientific community and the larger public community. And there's many, many examples I hope I've given you today of how the profile of the CVR has been enhanced by the public engagement work. And every now and again, a nice learned organisation will give you a prize and give you the opportunity to talk to large audiences about your public engagement work. So again, enhancing your profile and the profile of your institution. Finally, it's really enjoyable. All of the students who take part in the PCR workshop with me, they come away really enthused for public engagement. And I hope they'll carry that forward in their practice in the future when they go on to become program leaders. And I hope they'll be really active in doing outreach and public engagement, because it's such an important role for scientists to inspire the next generation of researchers and to inform the public about science and produce this scientifically literate public who are better able to engage with science information. So can outreach activities change lives? So this is a rather depressing quote from Carl Sagan about how some of us manage to retain our enthusiasm for science all the way through to having a career in science. But it's very true that children are born with this natural model building, experimental approach to life and learning about the world around them, whereas the formal education system can be very focused on knowledge acquisition rather than nurturing the sense of wonder and enthusiasm for discovery that children natively are born with. So I think outreach and engagement activities have this potential to reignite this enthusiasm. And I think probably everyone in this room will have a little story somewhere in their career about an inspirational teacher or an event that led to them, that inspired them to continue with their interest in science and to go on and have a career in science. For me, it was a field trip to a place called Sayers Croft, which is an ancient woodland field station in Surrey. So when I was a school student, we did this ecology field trip to Sayers Croft, and that exposure to real practical science really enthused me, and I was determined after that field trip that I was going to be an ecologist, and I went to university to be an ecologist. Here I am many years later as a structural molecular biologist standing in front of you talking about outreach activities. Clearly I didn't become an ecologist, but that exposure to practical science really fired me up and got me excited about being a scientist. And I'd just like to leave you with this idea that we all have this sort of story about the inspirational teacher or this sort of encounter that made us become a scientist, but you can actually become that inspirational teacher by doing activities such as these outreach projects, and you can enthuse the next generation of scientists to become enthusiastic about science and then consider a career in research. So I'm just going to finish up by acknowledging there are too many people to go through this whole list, but I'd particularly like to acknowledge Colin Loney, who's a research technician in my lab, who developed a lot of the reagents and the experiments that we use in these activities, and of course my wife Sharon McNabb, who has been a tremendous influence in my outreach activities. And I'm just going to stop with by showing you some web resources that you might be interested in looking at: molecular machines, the resources that we developed for the DNA applications activity workshop. If you're interested in the work in the Centre of Virus Research, you can like us on Facebook, and you can also have a look at this RC UK document about the benefits of public engagement. Thank you for your time.
Explore the fundamental role of microbes in the natural history of our planet with Microbes in Evolution, the world that Darwin never saw. Available at estore.asm.org.